All right, lecture number 7 for Unit 5, signaling between neurons. We're going to talk a little bit more, in a little bit more detail about what we mean by this signaling that's taking place when neurons network with each other. So some of this we've heard before. It gets into membrane potentials, those differences in voltage between, or the voltage, the difference in electrical charge between the inside and the outside of a neuron cell membrane. And there are two types of signals. Now we've heard about action potentials quite a bit in Unit 4. So that's the long distance change, temporary change in voltage across the membrane of the axon that travels all the way from one end to the other. It's an all or nothing type response. Once it happens, once it gets started at the axon hillock, it's going to travel all the way to the axon terminals, or those synaptic knobs. Graded potentials, those are local short distance signals. So those are local short distance changes in voltage. Those happen locally. And those are due to the influences in general, not always, but very often they're due to influences from other neurons. So on this diagram, you can see here there's a axon with an axon terminal from one neuron that's contacting that dendrite. Over here you see another one that's doing that. Over here you've got an axon of another neuron that's contacting that dendrite. So those axon terminals can secrete neurotransmitters, chemicals that travel across the synapse and cause what are known as graded potentials. Those are little local voltage changes. And if we're starting at minus 70 millivolts, like we talked about during Unit 4, those local changes, those local graded potentials, could cause that voltage to go lower, or it could cause the voltage to go higher. And so what you can have, like let's say this is a little area of stimulation over here, and maybe you got some stimulation going on over here as well. We'll say red represents stimulation, and then we'll say over here in blue, it doesn't have to be stimulatory, it might be inhibitory. So we might say in blue we've got some little local inhibition going on. Well, if those neurons keep trying to influence this one over and over and over again, you can have local stimulation over here and over here, local inhibition over there and over there. If you get more and more and more of it, these changes in voltage are going to approach the axon hillock. The axon hillock is our decision-making point. If we hit minus 55 threshold there, boom, we're going to get an action potential. So these local influences can add up and either make it more likely that we'll get an action potential generated or less likely. And it all winds up relating back to these voltage changes that are occurring. All right, and then once we get an action potential taking place, running down the axon. Some of this we've talked about before. There are a couple of things that influence that. Come on, pen, behave. One of those is myelination. So we've talked about that before. Unmyelinated axons conduction, that's the movement of the change in voltage, the action potential. Velocity refers to the speed. So in unmyelinated axons, that's going to be slower than if you have a myelinated axon with those little gaps, those nodes of Ranvier. That's called saltatory conduction, if you remember that from Unit 4, much faster in myelinated axons. Axon diameter, your larger diameter fibers, so if you're looking at them in cross-section, thicker ones are going to have faster conduction than thinner ones. Thinner fibers tend to have more resistance to flow of the action potential impulse. All right, and then your synapses between neurons. So we said we're trying to generate an action potential along the axon here. And a single neuron can receive influences or form synapses with many other neurons. And yes, very often another neuron is going to form synapses on the dendrites of a neuron. Those are called axodendritic synapses. Axo referring to axon of the other neuron, and dendrite tells you where the receiving portion of the other neuron is. Axodendritic. Axosomatic, remember the soma is the cell body, so this neuron, its little axon terminals are actually contacting the cell body itself, so they're called axosomatic synapses. And then in some cases the axon of another neuron will reach out and contact the axon hillock or even the axon of another neuron. Those are called axoaxonic synapses. And again, the influences of those other neurons, they might be stimulatory, they might be trying to stimulate this neuron, or they could be inhibitory, just like we were talking about with the reflexes. Sometimes interneurons will inhibit motor neurons rather than stimulating them. It's not always a stimulation type thing that we're talking about with the nervous system. All right, so when you have two neurons that are forming a synapse, the first neuron is called the presynaptic neuron. So that's the one that's trying to influence the next one. The second neuron in the line is called the postsynaptic neuron. So here's a synapse between two neurons. This one is your presynaptic cell, this one right here, and this one over here is your postsynaptic cell. So you're zooming in on the synapse that's occurring right there. You know, why do we have these synapses? If you didn't, then action potentials could just travel between every neuron that's networked with another one. Having the synapse there stops it and makes sure that you only transmit, you only continue this signaling if the next neuron receives enough stimulation. If all the neurons were just physically in contact with each other, then every time one neuron in your nervous system fired an action potential, that action potential would spread from cell membrane to cell membrane to cell membrane. There would be nothing to stop it. So that's why we have to have these synapses within the nervous system. The other things you see on here, the communication, you've got these chemicals that get secreted from these axon terminals. Those are called neurotransmitters. Acetylcholine was the example of that we saw during Unit 4. Those travel across the synapse and then they bind to receptors that are on the membrane of our postsynaptic neuron, and that's just like what we saw in the sarcolemma of our skeletal muscle fibers when they were being stimulated by acetylcholine from motor neurons. The rest of that story should look familiar to you here. You got the voltage-gated calcium channels that open up when an action potential travels down into this synaptic knob. Calcium flows in, tells these vesicles where the neurotransmitters are located to travel to the ends of the axon terminals and secrete those chemicals into the gap between the two. So that part of the story is just like what you learned in Unit 4. Yeah, and here's just another look at this. This is from your textbook. You got a presynaptic neuron that's influencing this one, and here's our postsynaptic neuron. So action potential comes down the presynaptic neuron, just like you learned about in the last unit. That triggers calcium channels to open. Calcium flows in. Calcium causes these vesicles where you have neurotransmitters stored to travel to the surface. They release their those chemicals, those neurotransmitters, into the synapse, into that synaptic cleft. Those diffuse across, bind to receptors, and those receptors, usually those are, once the neurotransmitter binds, those are usually ion channels that are going to cause some sort of a change in the charge across the cell membrane. It's not always going to be stimulatory. Sometimes, as we'll see, it's going to be inhibitory. And then that leads to, if you notice here on this diagram, a graded potential. So not an action potential at this point. When you have these synapses up on the dendrites or the cell bodies, you get little local changes in charge that take place. Those are called graded potentials, changes in voltage, local changes. The neurotransmitters. So as we learned about in Unit 4, you don't want these neurotransmitters hanging around here permanently. They are very temporary. That way, if you need to stop this signaling, you can stop it right away. If you want to continue it, you have more of the neurotransmitter being released very quickly. The way that those neurotransmitters are prevented from building up there in the synaptic cleft is they can be reuptaken. You have reuptake of the neurotransmitter. That's what they're showing you there. So they can be sucked back in to the axon terminal. Or you have enzymes in here like the acetylcholine esterase we learned about during Unit 4, which degrade those neurotransmitters. And a third thing is that they just simply diffuse away. Just simple diffusion. They're going to flow out of the synaptic cleft. And some of that again here is summarized on this particular slide. When you have a neurotransmitter, a chemical that's released from an axon terminal, it's only sitting there in the synapse for just a few milliseconds. It'll either be degraded by some enzyme that's located there, reuptake occurs. Sometimes it's those astrocytes. Remember those guys in the central nervous system? They're involved in reuptake as well. They might suck those excess neurotransmitters up, or they simply diffuse away. All right, so that leads us into, so we said this signaling between neurons can be excitatory or it could be inhibitory. Okay, all right, so we've got a diagram over here. We've got a presynaptic neuron. This is our presynaptic neuron. This is our receiving neuron or our postsynaptic neuron. This one has sent an action potential down. And if we look over here, that's triggering that neuron to release neurotransmitters. And those neurotransmitters are going to bind to receptors in the membrane of our postsynaptic neuron. Those are going to open ion channels, just like we learned back in Unit 4. All right, so if we're looking on our voltage graph here, let's say graph, not graft, we're starting out at minus 70. This is a typical motor neuron. And if the voltage change goes up like this, it becomes a little more positive than it was. That's excitatory. That's considered to be an excitatory change. Because if you have that, if you keep having that over and over and over and over and over again, those increased positive charges you have in that local area are going to diffuse and flow towards the axon terminal on this neuron. And if you get enough of that happening and your axon hillock there reaches threshold, boom, if you hit minus 55, boom, then this one is going to generate an action potential. All right, so if this presynaptic neuron, if it generates an action potential and that excites the receiving neuron, your postsynaptic neuron, that is called an EPSP, which stands for excitatory postsynaptic potential. Excitatory postsynaptic potential, that's an EPSP. And again, that's a local change like you see right there, but that local change makes it more likely you're going to wind up achieving threshold minus 55 millivolts at the axon terminal on this neuron. All right, then the opposite of that would be an inhibitory postsynaptic potential, an IPSP. So, as we talked about, sometimes you can have a neuron that generates an action potential, releases some chemical neurotransmitter, and that neurotransmitter binds to a receptor, which opens channels. Those channels, in this case, are generally for potassium ions or chloride ions that are negatively charged. All right, so let's think about if we've got our membrane voltage graph here. Keep wanting to say graphed today for some reason. We're starting out at minus 70 millivolts over here on our postsynaptic neuron. If you open up a potassium channel, that neurotransmitter binds to a receptor, and that receptor is a potassium channel that now opens up. Potassium ions flow out. Potassium ions are positively charged. What's going to happen to your membrane voltage? If positive charges are going out, then your local membrane voltage is going to go down. Okay, so that's called hyperpolarization. Remember, you're becoming overly polarized. Same deal too if that channel lets chloride ions that are negatively charged inside your postsynaptic neuron, inside this one. Same deal, your membrane voltage is going to become more negative. It's going to go even lower than minus 70. All right, and again, like we talked about, you get these little local changes happening here. Well, if that keeps happening over and over and over and over and over again, that's going to influence what's happening way over here at the axon terminal. So if the axon terminal becomes hyperpolarized, because this pesky little neuron here keeps sending these inhibitory neurotransmitters across the synapse there, this one becomes less likely to generate an action potential. It's going to be more difficult for it to do so. So those are called inhibitory synapses, and the voltage changes that occur along those synapses are called IPSPs, inhibitory postsynaptic potentials. All right, so the reality, though, is a single neuron is influenced by many others in general. Or a single neuron is not just going to receive one signal from another neuron that's trying to influence it. So like we're seeing here on this slide, a single EPSP, that's excitatory, cannot induce an action potential. Because if we've got our neuron over here, here's the axon hillock, here's the axon extending down. If you've got excitatory potential happening over there, that has to flow, it has to be conducted all the way to the axon hillock before the axon hillock is going to reach threshold, and you're going to have an action potential generated. But let's say you keep having excitatory, 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 excitatory. The inside in that local area becomes more and more and more positively charged. Those positive charge changes can eventually reach your axon hillock and cause you to reach threshold, minus 55. Or let's say you've got a synapse here that's exciting this neuron. You've got an excitatory synapse over here. You've got an excitatory synapse over here. And neurotransmitters are being released at all of those synapses at the same time. Well, those things can add together and influence what's happening over there at your axon hillock as well. So that's called summation. These EPSPs, those little local changes in charge, can add up. That's what that means. And cause you to reach threshold over here at your axon hillock. That's called summation. That can occur from the same presynaptic neuron. You can have one if it just keeps firing action potential, action potential, action potential, action potential. You're going to get enough stimulation of the postsynaptic neuron. Or the postsynaptic neuron might be receiving input from several presynaptic neurons. Likewise, IPSPs can also summate with each other or with EPSPs. So you might have a single neuron that's receiving 5 positive inputs from other neurons and say 3 negative inputs. Okay, so these guys are trying to undo the actions of these, and it's just kind of a matter of how things add up. If you add up to the point where you hit minus 55 millivolts at the axon hillock, boom, you're going to get that action potential. That's an all or none type thing once you hit the threshold. Or the inhibitory postsynaptic potentials might win out over the excitatory ones. Because a lot of times there's complex information that your brain is dealing with. Like if you see an explosion down the street, or well, let's not use an explosion. Let's say you hear a loud noise down the street. So you've had this auditory information that's coming from the ears to the brain. That's a danger signal. Maybe we need to run away. So you might have neurons that are processing that sound information, and there are neurons that receive that information up here. They send downward signals to motor neurons that are excitatory. Hey, we need to stimulate leg muscles to run. But then maybe you look over in the direction of the loud noise, and you're not seeing an explosion, you don't see any smoke, you don't see any other people panicking, you don't see an alien landing from a spaceship, anything like that. So you've got other information that's coming in. Maybe you don't smell anything unusual. And so that may cause other neurons to send inhibitory signals that are telling your motor neurons, hey, nah, we don't need to stimulate those leg muscles to contract, we don't need to run away. So sometimes you're dealing with competing bits of information as sensory input from your senses, and those things have to add up or subtract out to cause some sort of response to be made by your body. So that's just kind of a rough example of how a single motor neuron or sets of motor neurons that are controlling some of your skeletal muscles might be receiving both stimulatory and inhibitory information at the same time. And you can kind of see, well, if you're hearing something dangerous but none of your other senses are experiencing danger, then why waste energy and time running away from something like that? If we weren't capable of having these EPSPs and IPSPs competing with each other, we would constantly be making inappropriate responses if that was the case. Okay, so that just got into a little bit more of this signaling that takes place between neurons. I've been mentioning neurotransmitters so far throughout this unit, and the next couple of lectures we're going to talk a little bit more about neurotransmitters and what they are and the variety that we have within the nervous system, and we'll see which ones are excitatory, which ones are inhibitory. Some of them can be both depending on where they're used within the nervous system. So we'll start talking about those on the next video lecture.